My name is Erica Berg, and I'm the Director and Senior Editor of Custom Publishing here at Science. I'll be moderating today's webinar. Joining us today as guest speakers are Jeanette Bousbane from Stanford University, our grand prize winner of the 2025 Noster in Science Microbiome Prize, and Michael Fischbach, also of Stanford University, who we'll learn a bit more about shortly. Now, before I get started, I want to orient our viewers to what's currently being seen. At the top right of your screen, you'll find a photo of today's speakers and a View Bio link, which you can click on to read more about their background and research. After the speaker presentations, we'll have a short Q&A, which we will answer some of the questions submitted by our live online viewers. So start thinking about some questions now and submit them at any time by clicking the Ask a Question tab also on the right, typing the question into the message box and clicking Submit. And finally, a thank you to Noster for sponsoring today's webinar. Now let's begin. The skin microbiome is home to a diverse community of microbes that, far from being passive passengers, actively shape our biology. Recent discoveries have revealed that these commensal organisms are not only tolerated by the immune system, they engage with it in surprisingly specific and sophisticated ways. In today's webinar, we'll explore how skin-resident microbes influence immune responses both locally and systemically, challenging long-standing assumptions about how immunity is trained and maintained. We'll also highlight new research showing how our immune system builds a preemptive relationship with beneficial microbes and how these interactions may be harnessed for therapeutic innovation. So without further ado, I'm very pleased to introduce our speakers to you now. Dr. Jeanette Bousbane received her PhD from the Massachusetts Institute of Technology and is currently a postdoctoral fellow at Stanford University in the laboratory of Michael Fischbach. Her research combines bacterial genetics, immunology, and biochemistry to probe molecular interactions between the microbiome and the immune system, and leverages new mechanistic insights to develop non-invasive approaches to induce targeted immune responses. Dr. Michael Fischbach is the Liu Family Professor of Bioengineering at Stanford University, an Institute Scholar of Stanford ChemH, and the director of the Stanford Microbiome Therapies Initiative. Dr. Fischbach is a recipient of the NIH Director's Pioneer and New Innovator Awards, an HHMI Simons Faculty Scholars Award, a fellowship for science and engineering from the David and Lucille Packard Foundation, a medical research award from the Keck Foundation, and an Investigators in the Pathogenesis of Infectious Disease Award from the Burroughs Wellcome Fund. His laboratory studies the mechanisms of microbiome-host interactions. Wonderful to have you both here. Thank you so much for joining today. Jeanette and Michael will be tag teaming today's presentation, and I'll let them take it from here.
Fantastic. Thank you, Erica, for your introduction, and thank you for inviting us here. We're delighted to be sharing with you today. I'm going to jump right in on the next slide with the question that sort of frames the conversation today. I love the way you introduced the topic, but I'm going to move us back in time to the situation that Jeanette and I were in at the beginning, which was a lot more mysterious than where we ended up. And so let me frame it this way. If I asked you, what do you think the immune system is trying to do? What is it thinking? What's its purpose when it sees a pathogen, the situation on the left? We could discuss and debate, but my guess is that we would end up agreeing that the main purpose is to limit replication and dissemination so you don't die. But if I asked you, what do you think the immune system is thinking? What is it trying to do when it sees a microbial colonist in the gut or on the skin? I don't think you would know the answer. We certainly didn't. These are our friends, right? So it wouldn't be surprising if I told you that the immune system doesn't see them at all because there's a barrier in the way, or it does see them, but it recognizes them as friendly So it just gives a wave and walks in the other direction. But that's not at all what happens. So we're going to tell you what does happen, and try to convince you that it's bizarre, it sort of demands explanation, and could end up being useful. Okay, so let me explain some of the work, or summarize some of the work that had already happened by the time Jeanette and I got started, which is on the next slide. And I'll say it this way: certain commensals, when you put them on the skin or in the gut of a mouse, lead to a peculiar T cell response, a big burst of T cells on the other side of the barrier. The 4 characteristics of that response. The first is that the tone of the response seems to be dictated by the bug who's doing the inducing. So this ubiquitous skin colonist, Staph epidermidis, which colonizes every square centimeter of skin on every human on the planet, it will elicit a CD8 T cell response, whereas the gut bacterium SFB, segmented filamentous bacterium, will elicit a Th17 response, and a different gut bacterium, Helicobacter hepaticus, will induce a regulatory T cell response. Why it is that one bug induces one kind of T cell response, whereas another induces a different kind, is completely unknown. So I think the question that at the very beginning I thought that we were asking, but it turns out it's not the one that we answered. So this is still an open question. So the next slide will tell you the second odd characteristic, which is that the T cells that come out the other end of this process are antigen specific, meaning that they express a T cell receptor that is hunting for a peptide on the bug who did the inducing. This is not a simple matter of bugs kind of turning up or down the overall dial on the immune response. This is a highly specific response of the sort that you wouldn't be surprised to see if the immune system were going after a pathogen. But here, remember, it's in the context of the response to a colonist. So in the next slide, the third strange characteristic, which is that textbook immunology would have it that if you're going to get a big adaptive immune response, the situation on the left, there is a breach in the barrier, either an infection event or a vaccination with a needle going through the skin. 5-alarm fire, innate immune cells rush in, there's a lot of swelling, and hopefully that innate immune response summons an adaptive immune response, which clears the infection, leaves you with immune memory. By contrast, the situation on the right seems completely different. You put a microbe on one side of the barrier, and apparently nothing happens. If you look in the tissue, there's no swelling, no inflammation, no sign that anything immunologic is taking place. It's just that 7 to 10 days later, a big burst of T cells shows up on the other side. And even more puzzlingly, on the next slide, they don't seem to have any clear function. They largely will just loiter near the site of colonization, not do much of anything. So this is the puzzle that we walked into. As many of our listeners will appreciate, Jeanette and I agreed that the immune system, if anything, doesn't trifle. It doesn't sort of act without purpose. And so this is an odd situation where we seem to be mounting a big immune response that doesn't have a purpose and no apparent one. And so we thought that this didn't add up and wanted to learn more. There's one piece of data that we had that kind of informed our hypothesis here, and it was gathered on the next slide by Jeanette's former lab mate, Erin Chen, who kind of got this line of work started in my lab but before she left to start her own lab. And so I'm summarizing what she did on the next 2 slides. I'm going to explain the experiment she performed. I'm going to say what she did and then explain why she did it. Sort of makes more sense in that order. So Erin had developed a genetic system that made it possible to insert transgenes into Staph epidermidis. And she used that system to put a tumor antigen on the surface of the bug. Why did she do that? Because she wanted to trick the immune system into developing T cells that it thinks are specific for the bug, but we know would also have specificity for a tumor that resides elsewhere in the mouse. Why did she want to do that? Because it would create a useful functional test for those T cells. 2 diametrically opposed possibilities. The first is that they would just do what everybody else had observed them to do, which is to sit near the site of colonization and do pretty much nothing. The other possibility, of course, is that they would do what any self-respecting CD8 T cells should do, which is to leave the skin, go to the tumor, and kill tumor cells. So this was really a test of what their functional capacity was. The way the experiment went is summarized on the next slide. So a very, very simple experiment. This takes place in conventionally colonized black 6 mice. These are wild type mice. They have a competent immune system, and we inject in their flank a syngeneic tumor cell line, so same genetic background as the host, and it grows as a primary tumor mass sort of underneath the skin on the hip of the mouse. We don't do any preparation. There's no shaving or scrubbing. All Aaron did is to dip a Q-tip in a bacterial culture and rub it gently on the head of the mouse. That's the whole experiment. If we click to the next slide and summarize what happened, if you don't treat the mice at all, that's the naive group, or you treat them with a wild type strain of Staph epidermidis that doesn't express a transgene, all the tumors will grow out of control and all the mice will end up dying. But in the group that was treated with the test article, which expressed 2 different forms of ovalbumin as a tumor antigen, the tumors were gone. And so there's a lot to say about this, and that's not the topic of today's webinar, so we won't go through it here, but I will simply say, as I hand over to Jeanette, that Jeanette was helping Aaron with this project, and she and I were both at the same time devising the question that she would ask, and I think that we were all quite surprised at how aggressive this immune response was compared to our expectation that it would be gentle and soft and homeostatic and there to keep the peace. And so that was sort of the state of mind that we were in at the very beginning of Jeanette's work. And now over to her.
Yes, thank you, Michael. So seeing the response that we saw here against tumors, we wondered, is the response more similar to a response to a pathogen that we had anticipated? And if this is the case, wouldn't we expect to see engagement of both arms of the adaptive immune system, and in this case, B-cell responses? And so this is a pretty basic question. Is there a B-cell response against skin microbes? You would think that someone would have looked into this, but the answer is that it wasn't really studied before. The best evidence in the field was from this paper in 1991 that took swabs of human skin. And as you can see here in electron microscopy, certain microbes that are present on the skin are coated with different types of antibodies of different isotypes, suggesting that 1, these microbes induce a B-cell response, 2, that antibodies are secreted on the surface of the skin, and 3, that by coating these microbes, they may influence the behavior of these microbes. A second piece of evidence came from a paper about a decade ago. For a long time, people believed that B cells were devoid, the skin was devoid of B cells in the skin of healthy tissue. And so it was actually shown that in a healthy, non-inflamed skin, you could find B cells. In addition to that, it was also found that not only B cells were present, but antibody-secreting cells. And these cells were accumulating over time, suggesting that maybe they recognized an antigen that accumulated over time as well, just like a microbial-derived antigen. And so we just asked the question, if we start with our favorite bacteria, Staphylococcus epidermidis, and we colonize its skin, just like the Belkaid lab or Erin Chen had done it, by dipping a cotton swab into an overnight culture and gently rubbing their head, can we find the presence of antibodies specific for Staph epi systemically and locally? And what we found was actually quite remarkable. As Michael mentioned, we identified the presence of very large titers of antibodies specific for Staph epi in the serum of mice. This response was also quite interesting because instead of going up and down, just like you would see for like a vaccination, it gently go up and then it would stably stay there and plateaued around 6 weeks. It almost felt like the host was kind of vaccinating itself against its own microbe. We also could identify antibodies specific for Staph epi in the nasal wash locally, and that's because this microbe can also be found at the entrance of the nose. Next, we asked whether it could induce the germinal center reaction in the draining lymph nodes and affinity maturation. So mice were colonized with either Staph epi or another skin commensal C. accolans that is known to induce gamma delta T cells. And what we found is after 2 weeks of colonization, Staph epi could induce the generation of germinal center B cells, but not C. accolans, suggesting that it was also inducing affinity maturation. Next, we asked whether the response was strain-specific. So on your left, you have a phylogenetic tree showing you a number of different Staph epi strains, LMO LM61 all the way to LM195, and then our control strain C. accolens. Mice were colonized with one of these different strains, and then we looked for the presence of antibodies specific for any of these strains after 5 weeks. And as you can see here in the heat map, on the top you have the mice colonized and then the strains, and then on the left you have the response too. So the first thing that you can see is that mice colonized with C. accolens, our negative control strain, do not have antibodies against C. accolens. In contrast, you see variable levels of titers when you colonize with different strains of staph epi. The best and strongest response was when we colonized mice with the strain LMO88, and you can see it's very specific to this strain and not any other staph epi strains. In contrast, the strains that are shown here in purple are part of the same phylogenetic clade, and you can colonize with any of them and you can get cross-reactivity to the other strain of the same clade. And so from this, we can conclude that the response is strain-specific at the exception of strains that are derived from the same clade. So one of the things that was raised was the question that maybe the way we colonize mice could be artificial, right? Because we are providing a high amount of bacteria on their skin. And we wondered, is this response that we are seeing just due to the fact that we're actually providing so much signals, so much antigens all at once, that we see such a strong response. And so what we did is take SPF mice, so standard pathogen-free mice, that are naturally colonized with a number of skin microbes, and we asked the question, can we now take their serum and look for antibodies specific for them? And that would suggest that the response is conserved in mice and that it's not due to the fact that we are spreading high amounts of bacteria on their skin. And the answer is that yes, this response was conserved. We could isolate 2 different of these microbes, a Staphylococcus psilocys in Jackson mice and a Streptococcus species in Taconic GT mice. And you can see that these responses are specific to the mice that actually harbor these microbes. So the Staphylococcus psilocys strain elicits a response in Jackson mice, but the other mice housed in different facilities do not have this bug and do not have responses against it. And the same is correct for the Streptococcus strain. And perhaps even more interestingly, you can also find microbes present on the skin that are there. They colonize very efficiently these mice, but they do not induce a response. And so some microbes present on the skin are able to induce a response. This response is specific, but not all of them do. We also asked whether this response was limited to mice or was actually conserved in humans. So we took the serum of 11 healthy donors and we looked for the presence of antibodies specific for microbes that are typically found on human skin. You have here on your left, you can see a range of different staph epi strains, other Staphylococcus and Corneobacteria. And you can see that there are very high titers of antibodies in their serum specific for these different microbes, especially for Staphylococcus and to a lower extent to Corneobacterium. And on your right, you can see the plots showing the titers per donor. And that shows you that this is not one specific microbe inducing a response, and that's always the same across donors. It's like each single donor has its own flavor of responses to these microbes. Next, we were interested in identifying the actual molecular determinants of these interactions, meaning the antigens that are recognized by the response. On your left on this slide, you can find staph epi coated with antibodies that are raised against itself. It's derived from the serum of mice colonized. And you can see that very efficiently, there's a lot of these little black dots, and those represent coating of antibodies to its surface. From this, you can guess that there is an antigen present on its surface. And so what are the antigens present on the surface of staph epi, and how can we figure out what is the antigen that is actually recognized? On your right, you have a cartoon depicting the main structures found on the surface of staph epi and more broadly on the surface of gram-positive bacteria. You have teichoic acid that are lipid-anchored or wall-anchored. You have wall-attached proteins and lipoproteins. So how could we distinguish which of these structures was recognized? I was in luck because my labmate Erin Shen had made a library of cell surface mutants. What she did was to mutate one gene at a time, and these genes were important for each of these different structures. And that allowed me to identify among these different structures which one was recognized by the antibody response to Staph epi. So from left to right, you have the wild-type strain. Then you have a delta LGT that has no lipoprotein, a DDLTA that has no D-alanine at the end of the teichoic acids, a D-sortase that has no cell-attached proteins, and a D-tago that has no wall teichoic acid. And then each of these different mutant strains were coded on an ELISA plate, and then we tried to see whether the antibody raised against the wild-type strain could still bind these different mutant strains. And what we found is represented here on your right. The bacteria that had no sortase, the sortase mutant, had a reduced binding to the antibody specific for Staph epi, suggesting that a cell wall attached proteins was among the antigens recognized by the immune system. What is sortase? Sortase is a highly conserved enzyme present across gram-positive bacteria. And what it does is that it recognizes a conserved sequence at the C-terminus of proteins that need to be attached to the cell wall. And you can see here, it's written here as LPXTG. And so from a whole genome, we were able to only look at about a dozen proteins that had a LPXTG motif to find the antigen recognized. We had a second piece of evidence, and I'm showing this here. So we looked at the sortase substrate present in two different Staph epi strains, LMO87 and LMO88. And we are representing them here based on their size and whether they're present in both strains or only one strain. So in gray, you have the strains, the sortase substrate present in both strains. In blue, only in LMO88, and in purple, in only LMO87. And on your right, you have a Western blot analysis showing the lysate of LMO87 or the culture supernatant of 87 or 88 next to each other. And then these bacterial lysates and supernatants were blotted with the serum of mice colonized with LMO88. What you can see here is that there are common bands between the two strains of bacteria, for example, at 37 kilodalton. But in LMO88, you have these additional high molecular weight bands that are only present in LMO88 lysate, but not in LMO87. If you are looking then on your left with the different sortase substrates, you can see that really there's one protein that is big enough, AAP, to be able to be recognized here at above 150 kilodalton. So we generated a knockout and a complement. So you can see here, it's the same analysis, a Western blot analysis with the lysate of 87, 88, LMO88 with a knockout for AAP, and then a knockout of AAP with the complement expression of AAP on a plasmid. And you can see very nicely that these high molecular weight bands, they are present in 88, absent in the knockout, and they reappear in the complement, suggesting that indeed AAP is an antigen present on the surface of Staph epi. So what is AAP? AAP stands for accumulation-associated protein. It's a very interesting protein. It's very big, and it's composed almost exclusively of repeats, either sequence repeats or structural repeats. So from the bottom, the C-terminus, you have a B domain, which is also composed of repeats, G5 and E spacers. And on your right, you can see the predicted structures. They form these beta sheet sticks that makes the protein extend far away from the surface of the bacteria. And we think that this is important for recognition by the immune system. The next domain is a parallel beta helix that is big and globular. And then you have amino acid repeats at the N-terminus with undefined structures. When we mapped the epitope, the regions that were recognized by the antibody response, we could see that the main antigen was present in the B domain, but that the parallel beta helix was also recognized. So from this, if we summarize what we have found, when we colonize mice with wild-type Staph epi, we can find B cells and antibodies specific for Staph epi. But what's the purpose of this response? So, seeing the magnitude of the response and how much antibodies were raised systemically against Staph epi, we kind of wondered, would it be possible that the purpose of commensal specific immunity be there to protect the hosts? Are we kind of constantly vaccinating ourselves against our own microbe preemptively? And that would be because, as you can imagine, these microbes live so closely with us. Sometimes they're separated from us by a single layer of epithelial cells. And so a little cut on your skin is inevitable, and these microbes can breach the barrier. And if this is true, wouldn't you want to have immune cells present at the site of colonization, ready to respond right away and prevent further spreading? And if this hypothesis was true, could you imagine engineering this response and creating new forms of vaccination? And this is what we asked. So the idea here is, can you take Staph epi and express a little piece of pathogen, this orange pathogen, and then colonize the skin of mice with these engineered microbes? And can you then induce immunity specifically against this orange pathogen? And then can you challenge these colonized mice with the pathogen and see if they are protected? And this is what we are showing here. Can we express on your left a non-native antigen, the orange antigen, and then induce antibodies against it? How would you go about this? Well, it's not as simple as just expressing an antigen on the surface of the bacteria and colonizing and getting a response. What we had to do was actually go back to what we knew against the native response. We knew that AAP was recognized, and we knew that different portions of the proteins were recognized. And so we hypothesized that by flipping part of this protein, switching a domain of AAP with a new antigen, we could actually induce a response against both some other parts of AAP, but also this non-native new antigen that was derived from a pathogen. So to test this hypothesis, we took advantage of the tetanus toxin model. So tetanus toxin has a fragment that is non-toxic. It's called tetanus toxin fragment C, or TTFC. And so we generated strains which either expressed a secreted version of tetanus toxin fragment or a fusion between AAP and TTFC. And I'm showing them here. And we asked, if we colonize mice for 6 weeks, can we actually find antibodies specific for this new antigen in mice? And the answer was yes. So systemically, you can find antibodies specific for TTFC when you colonize with the AAP TTFC fusion. You can also see it to a lower extent when you colonize with the secreted TTFC systemically. However, locally in the nasal wash, you can only see IgA specific for TTFC when you colonize with the AAP TTFC fusion. And we wanted to make sure that this response was not limited to the antigen that we worked with. And so we generated a catalytic mutant version of diphtheria toxin. And we could show that, again, mice colonized with this catalytic mutant version of diphtheria toxin could induce a response against diphtheria toxin, showing that not just one antigen, but 2 antigens worked. Next, we asked the question, okay, so we're seeing an antibody response, but now what does it mean? Is it functionally relevant? Are we getting neutralizing titers? Are these titers high enough to actually protect against a challenge? And so we colonized the mice with either of these 2 AAP fusions, so either to TTFC or the diphtheria toxin mutant, and then we challenged these mice with a lethal dose of tetanus toxin. This is a very acute model, and mice develop pathology in about 2 days. And this is what we observed. On your left, you can see mice that were colonized with the control strain, the diphtheria toxin strain, and then injected with the lethal dose of tetanus toxin. So about 2 days later, you can see they are having issues breathing. They're actually developing a paralysis that is typical of a tetanus disease. In the middle, you have the mice that were colonized with the tetanus toxin strain. And you can see that they're totally fine, they're running around, and they do not show any sorts of pathology. And they actually remained healthy until the end of the experiment. This is the same mice that I'm showing in the middle, you can see here, at over 200 hours. And here, I'm just showing you the quantification for the different mice. It's basically the same as the video, but for more mice. All the mice that received the tetanus toxin strain are fully protected for the whole experiment, and all the mice that received the control strain developed tetanus. Next, we asked whether this protection was mediated by antibodies. And so to do this, we went back to the mice that were colonized with either the tetanus toxin strain or the diphtheria toxin strain for 6 weeks. We took the serum of these mice, and then we mixed that with tetanus toxin and injected that into a new recipient mice. And then we asked the question, is there antibodies in the serum that are capable of neutralizing the effect of the toxin? And the answer is yes. You can see again here that all the mice that received the serum from the mice colonized with the tetanus toxin strain were fully protected against tetanus toxin, but not the control serum. And so from this, we can conclude that not only we were able to induce a strong response, redirect the response against a new antigen, but this response was strong enough to protect against a lethal dose of neurotoxin. Building onto these findings, we wondered, what if we wanted to do something similar, but with an antigen that we cannot express in bacteria? Think about a viral antigen. Viral antigens form big complexes. They have disulfide bonds. They have glycosylation. All of these things are very difficult to express in bacteria. And so could we actually conjugate or label the surface of the bacteria and then colonize mice with these bacterial labeled strains and induce a response against this new antigen? And so we made 2 different assumptions here. One was, can we actually conjugate antigen to the surface of the bacteria? And 2, can this pulse of antigen be sufficient to actually induce a response in vivo? First come first, how do you conjugate antigens to the surface of a commensal? We took advantage of the SpyCatcher system. SpyCatcher is actually a system derived from a sorted substrate from a different gram-positive bacteria, and 2 different pieces of this protein was split into 2 different tags, a SpyCatcher tag and a Spy tag, and you can hook that up to 2 different proteins. And when you mix these 2 different proteins, these 2 tags will spontaneously react with each other and form a covalent linkage, and this reaction is irreversible. And so we equipped AAP with the SpyCatcher motif, and then we mixed these engineered strains with an antigen that contained a Spy tag. Then we asked whether we could decorate the surface of Staph epi with this new orange antigen. The answer is yes. So here I'm showing you 2 different strains, a SpyCatcher catalytic mutant, so it cannot react with Spy tag or much less, and then a full AAP SpyCatcher strain. And then we had a GFP attached with a Spy tag. And you can see here on your right that the strain that has a functional SpyCatcher can bind very efficiently to GFP, but not the catalytic mutant. And so the first point, yes, we were able to attach antigens or protein to the surface of Staph epi in vitro. Next, we asked this next question, which was, can we actually induce a response with these conjugated bug in vivo by providing a pulse of antigen? Mice were colonized with either the naked SpyCatcher strain, so there's no antigen, a SpyCatcher strain conjugated to the tetanus toxin fragment, or our genetic fusion as a point of comparison. In addition to this, we wanted to see how our strategy performed in comparison to a more conventional approach. So we use Mi3 nanoparticles, which are SpyCatcher-based nanoparticles that have up to 60 binding sites for antigens, in this case TTFC. And we immunize a group of mice with these nanoparticles in a typical prime boost approach. And then we ask the question, how does our strategy compare to this more conventional approach? As you can see here in the serum of mice, both our commensal approach gave good, very high titers against tetanus toxin. And remarkably, these titers were, although slightly lower than the nanoparticle injected, the order of magnitude was similar to what we saw with a very potent injected approach. Perhaps even more interesting if you now look in the pulmonary mucosa, in the nasal wash or the bronchoalveolar lavage, you can see that both commensal strategies induce good IgA responses against tetanus toxin, unlike the nanoparticle injected strategy that gives very good systemic response, but very poor mucosal immunity. And this is quite interesting because, as you know, a number of conventional vaccines are very good at inducing systemic response, but they are not so good at inducing local response, which are essential to block the entrance of pathogens because a lot of pathogens enter through mucosal surfaces. And so if we were able to induce both systemic and mucosal response, we would hopefully be able to not only protect against disease, but also protect against infection and transmission. Another point about this strategy was that this response was long-lasting. So we colonized mice for our typical 66 weeks, and then we look up to 6 months, so about 4 months later, and you can see that we can find titers against tetanus toxin all the way to 6 months, and there's barely any decrease. And so this response is not only of a high magnitude, but it's also very long-lasting. Next, we wanted to ask, what is the limit of this strategy? So as you can see at the top, we colonize many times, 13 times. We do either 8 or 13 times most of the time. And so we asked, can we actually reduce the number of colonization to something that would be more similar to what you would get for a vaccine, which is like a prime boost? And so can we reduce the number of colonization to 6, 5, or even 2 and still see protection? So first, looking at titers, systemic titers against tetanus toxin, you can see that only 2 colonization did give titers against tetanus toxin, although 2 with more variation. You see that there's more variation between mice. But in average, you see that by increasing the number of colonization, you reduce the variation and you slightly increase the average, but not very much. It's mostly the variation that is reduced. Next, we asked whether the mice that were colonized only twice could be protected against a lethal dose of tetanus toxin. And this is what we are showing here. You can see on the bottom right, the mice that were colonized only twice, most of them were again fully protected against a lethal dose of tetanus toxin. And this is quite remarkable because, as you can imagine, only 2 colonization is very little to get such a strong response. And so to conclude, I hope that I've convinced you that Staphylococcus epidermidis and other skin commensals are capable of inducing B-cell responses that are both systemic and local. And by identifying the main antigen that is recognized, we were able to redirect the response and create new forms of topical vaccination.
Great. So to pick up from where Jeanette left off and to wrap up the presentation, let's go back to the question that we started with, which is what have we learned about the purpose of the immune response against commensals? I don't want to overgeneralize here, but at least in the case of Staph epidermidis, it's not at all what we thought. This is not a response that's simply meant to maintain homeostasis. It's nothing gentle. It's certainly not local. In almost every way, clicking forward to the next slide, this response mirrors what you would see in the response to a pathogen. It's quite aggressive, both the T cell component and the antibody component. It's broad spectrum, involving a pretty high octane adaptive immune response. Really, the major distinguishing factor is that this is happening preemptively. This is not in response to an event. This is happening while you're well. And I think that raises really interesting mechanistic questions, which will be on my very last slide. So clicking forward once, there's two thoughts to leave you with. Well, one slide yeah, there we go. Jeanette and I are quite interested in the possibility that this could create a new kind of vaccine. Of course, there'd be no needle and no healthcare worker. This could be applied topically. You could apply it to yourself in the form of a cream, could arrive in the mail. So I think that would have a lot of advantages logistically and in terms of the ease of distribution. Something that we're puzzling over, but seems to be quite consistent, is that there are no signs of reactogenicity. So this could be, if this continues to bear out in larger mammals, a sort of vaccine that could elicit a strong adaptive immune response, but without making you feel lousy, which I think would remove a major barrier to vaccination that currently exists. as Jeanette pointed out, not only are we seeing a high titer of IgG systemically, but we're also seeing IgA in the mucosal surfaces of the nostrils and lungs. And we didn't mention in the talk, but it's also present in the small and the large intestine. So the mucosal antibody response could make this useful for blocking transmission of a respiratory pathogen or for pathogens that infect at the gastrointestinal mucosal surface. And your skin is a large organ. So where you would never bring a kid or even an adult into the doctor's office for 6 shots, you could definitely rub a tab of cream one behind each ear and one on each elbow and one behind each knee, which would all drain to different lymph nodes. And so you could imagine having a multiplex vaccine against multiple pathogens. That would be straightforward to do in this format, which is something that we're quite excited about. So on the final slide, leaving you with this thought, which may help seed some questions for the Q&A, we're quite interested following up in several different things. I'll highlight this question, which is how does this work? What is the mechanism here? We think there is the possibility that some cell in the immune system, some innate immune cell, possibly the Langerhans cell, evidence for which was gathered by our collaborator Yasmin Belkade and published in the paper that accompanied Jeanette's, reaches across the surface into the outside world, grabs antigen, and brings it back inside. If this is true, a couple things. Well, one is that would be fascinating from a basic science perspective. The second is it could be quite useful. And the third is we need to know exactly how this works, what cell is doing this under which circumstances, and what exactly is grabbed and how is it brought in and what is its fate once it comes back inside. So this is a question we're quite interested in and working hard on. So in our final slide, I want to acknowledge Jeanette and the team in the lab that worked with her, along with our funders in the upper right-hand corner. And thank you all for listening.